Hello, everybody. It is good to see you here in person and the many people who are online for our second ever Black History Month Neuroscience seminar. I have the pleasure of introducing Dr. Kacie Deters, who is a new faculty at UCLA, and she's been here since not even a year. Yeah, July of last year. Dr. Deters did her postdocs at Stanford and at UCSD, where she did research on risk factors for Alzheimer's disease based on sex and race. She did her bachelor's degree at the University of Alabama in Birmingham, and then a master's degree in biology at Cal State University Dominguez Hills, which is exciting to me because I have another wonderful student from that same place. And she did her Ph.D. in medical neuroscience at Indiana University. She was our hottest recruit, and we are so excited to invite her to give this second annual lecture. Thank you. Please welcome Dr. Deters.
Thank you, Gina. That was unbelievably sweet. Let me just get rid of this floating meeting. All right, hi, everyone. Thank you so much for the warm welcome. I'm very happy to give my first in-person lecture at UCLA as faculty. So thank you all for being here. As Dr. Poe mentioned, I'm going to be talking about examination of well-established risk factors for cognitive decline and dementia in the Black community. But more importantly, I'm also going to be talking a little bit about Black History Month. Oh, there we go. Okay. So Black History Month to me is Black excellence. And so that's exactly what I'm going to highlight in this talk today. Black excellence, both past and future, or in present. So I'm going to start right here at UCLA with the Ralph J. Bunche Center for African American Studies. I really love this picture highlighting Dr. Bunche here meeting MLK and his wife. So Bunche won the Nobel Peace Prize in 1950. He was the first African American to do so and the first person of color overall to win. Sorry, these floating meetings just keep popping up. Sorry, I don't know how to make it stay on. Oh. I'm just going to move it to the top and then it shouldn't issue.
Just on yours?
Yeah, it goes away. Should be okay.
Move it to the very.
Yeah, it's not. I kept doing hide and then it.
Let me see from out there if I can disable it entirely.
Thank you. Sorry about that. So yeah, he also graduated UCLA summa cum laude and pop right back up over where I need. I'm just going to move it to the top. Okay, sorry. In 1927. And I learned about him when I was actually interviewing for this position. Not only was a job call related to a movement here at UCLA to recruit more Black and faculty and staff and graduate students, but also my room happened to be on the same floor that his picture was hanging. So it was really interesting that every time I walked out of my door while I was interviewing for this position, I got to see his face, and it was a little bit of like a motivator for what I was doing. So then in 2003, the UCLA Center for African American Studies was renamed after Ralph J. Bunche. So as I mentioned, this actually led to me getting my job here. There was a UCLA-wide initiative called Rising to the Challenge that was devoted to advancing racial justice and Black studies here. And this email just went out to, I'm not sure who all got it, but I got it via UCLA. And out of the 10 faculty that they plan to hire, there's at least 7 hires and across UCLA. So you can see from law, public health, public affairs, neurobiology, integrative biology, and physiology, which is myself, and all focusing on different aspects of Black life. So this has led me, I've been unbelievably happy here at UCLA so far, not only with the department, but with, this recruitment of other Black faculty. I've been able to connect with a lot of peers that are going through similar things, such navigating new faculty as a Black person here on campus. And a lot of people don't have that. I do feel very, very fortunate that I do have like a cohort of people who look like me. And through this cohort, I was able to meet this gentleman here. This is Dr. Justin Dunavant. I hope I'm pronouncing his name right. He's in the Department of Archaeology. He's been here about 2 years, and he organized a wonderful tour last Friday for the Kinsey collection, which focuses on African-American art and history, and it's currently at the SoFi Stadium right now. We were so lucky to have the Kinsey family actually give this tour. It was unbelievable, the lengths that these people have gone to protect Black history and art. They did not come from wealth, the Kinsey family. They accumulated this over time themselves when people were not names, were not big names. So a lot of the art that they collected, that's worth multi-millions now, was not when they initially bought it. And I wanted to focus these 2 pictures here. The first one, in the background, this is a painting done by Samuel Dunson that is actually of the Kinsey family. This is supposed to be representative of the dad, who is collecting books. The mom is washing books, and the son is reading the book. And I just thought it was really, really, this whole thing was touching. It was actually very hard not to cry when walking through this show, the collection. They also have a lot of, art pieces, historical pieces. So for example, slave letters, slave documentation, the bill for people of sale. They've collected everything that's historical and cultural. And on top of that, they really wanted me to mention that today is their anniversary, 56 years. This is the painting of theirs in the background. They're just like the cutest couple ever. They were like, I said, oh, can I get a picture of you? And they're like, yeah, come in front of our photo though, so we can reenact it. And it was just super cute. Anyway, they have about 10% of their entire collection right now down at SoFi. And so, yeah, I really suggest that you go and, if you can, take a tour. And after taking the tour Friday, when I was talking with the family, I said I wanted to highlight it in this. And he said, make sure you show this. This is the most important thing. And so I'm just going to read the first part of the paragraph that he really wanted me to bring attention. In American history, as in American life, Black Americans are invisible presences. They are not seen, not because of their absence, but because of the presence of a myth that prepares and requires their absence. The myth of absence, which expresses this idea and intention, operates not by misinterpretation and slander, but by silence and exclusion. Very powerful. And I think it was, he also gave, Mr. Kinsey gave a really great example of how this applies to Black life as well. He said it's like navigating a cemetery with no headstones, trying to find our history and our culture. So I also wanted to highlight Black and Neuro, of course. Black and Neuro started just a few years ago, and it's been an amazing way for Black folk who are neuroscientists to get together. There's a lot of Black and X at this point, Black in genetics, Black in chemistry, so forth and so forth. They have a presence on Facebook, YouTube. There's a lot of seminars that are recorded, for example, and uploaded to YouTube, Instagram, Twitter. And the Impact at a Glance, they do a yearly update. And so this is just what's happened in the past year. 540 members, 27 events, a whole lot of Twitter impressions and engagements, and a lot of event registrations. And I just wanted to share some of these events that are happening in Black and Neuro. We have a monthly seminar series, which I'm the lead on. I have like very different pictures throughout this, you'll see. But I'm the seminar series program lead for this year. Somebody was probably taking over at the end of this year. There's been pals to talk with, like to say baby scientists. There's a lot of socials, professional development, perspective blogs, and highlighting scientists for Black History Month. And the socials has been obviously hard to do in the middle of COVID. This actually started in COVID. And I wanted to highlight what happened at Society for Neuroscience meeting this past year. This is just amazing to see so many Black neuroscientists, Black scientists together in one space. And if I remember correctly, the picture on the left are Ph.D. students. The top picture are postdocs, and the bottom picture is faculty. And it's important to note that this is not it, right? I know Dr. Poe, Dr. Kacie was at SFN, but unable to join. There's a lot of a lot more people here that are present, but it's nice that we had a place to connect. Also, being a person of color in science is not easy. There are a lot of challenges, and I'll always, typically I put this slide at the end during my acknowledgements, but I'm going to put it up front for now. If it weren't for all these people pictured here, I don't think that I would be where I am today. There were a lot of times that I wanted to leave science. There was just a lot of hard things that I went through personally, and all of these people pictured here have helped me one way, whether it's just allowing me to complain to them or they helped me with job prospects and so forth. So I really like to acknowledge every time I give a talk now, I call them my mental health and academic warriors. And again, I know there's trainees, whether they're in the room or online. I also like to cover a little bit, if I can, during every talk where there's trainees present, my trajectory, because my trajectory was not normal. It took a lot to get here. And so I kind of put my timeline up here just to talk about all the struggles of how hard it was for me to actually get here. I come from a single parent household. I'm first gen, grew up in an apartment, borderline low to middle socioeconomic class. So I really did college on my own. I had no idea actually what I was doing. I just knew you're supposed to go to college. And so I went as far away from home, I'm from California, as I could, of a place that I got in, and that was University of Alabama, Birmingham, as Dr. Poe mentioned. I actually failed 3 classes. I had no research experience during this time because I had no idea what I was doing in college. Massive student loans, very low GPA I graduated undergrad with, if you can see there. And a lot of that, though, is due to not only me not just having the mentorship that I needed at that time, but also I had to work 3 jobs. I worked on average 7 days a week up until my last semester because I was just tired. And then I actually cut down to 6 days a week, which is still a lot, but you're young, have energy, I feel like at that time. No, I did not work at the Gap. The Gap just represents gap year. I took a year off. Also worked 3 jobs. Again, not at the Gap, but worked 3 jobs, tried to figure out what I should do with my life. And I was like, okay, why don't you just get a master's degree and see if you can do this? So again, very low GPA. I was not going to get into, and no research experience. I was not going to get into a top-notch place like UCLA. So I got into Cal State Dominguez Hills, where I worked 2, I actually started out with 3, and I was like, you can't do this again, Kacie. Dropped down to 2 jobs, picked up my GPA, and I was very fortunate to get a grant through Cal State Dominguez Hills, who did a great job at really recognizing the importance of bringing research to people who might not have it. And I got this bridge to the doctorate grant, which paired me with Indiana University to do summer research. So that's where I kind of got my first research exposure. Actually, it was in the middle of my master's program. And it may not look like there's a gap because of the year, but I actually did take a gap year because, again, I was like, okay, do I want to do this full time? I actually moved to Dallas, Texas randomly and just lived and worked. I worked 2 jobs, 6 days a week, about 12 hours a day during those times. Then I got into the PhD, and finally, I'm not working any jobs because, as you know, a PhD is a full-time job. I actually failed a class in my PhD, and I failed my qualifying exam. It is still possible to make it even if you fail. I always like to give this message because I failed a lot along the way. I am just not a classroom person. I really hope I can change that in the future now that I'm a professor myself, but I just really like to send that message if I can. And you can see that classes don't equivalent success. Or I still had 4 first author publications. I got an NIH grant. I got another fellowship, and I had several travel awards, poster presentations. And so that well prepared me for my postdoc at Stanford. Then COVID hit, didn't want to start a lab or anything during COVID. Went to UCSD for another short postdoc. Applied for a KO1, just found out like last Friday or 2 Fridays ago, my KO1 was funded. So yay. And now I have some funding as professor. Thank you. Very excited. And now I'm here at UCLA, and I'm very excited to be here. So I do like to talk about this because Black excellence is not just one thing or another. It's not just, it's continual work that we have to do in this space. So I'm going to take a sip of water, and then finally we can get to some research. So Alzheimer's disease is the most common form of dementia. There are 2 main pathological features that are present in people with Alzheimer's, and that is beta amyloid plaques and tau tangles. So on the left side, you can see a cognitively normal, healthy brain post-mortem. And on the right side is somebody who has advanced Alzheimer's disease, in which there's a lot of atrophy or shrinking of the brain, especially in this area here, the medial temporal lobe, where the hippocampus is. Alzheimer's disease and other forms of dementia are marked by cognitive decline. There is some cognitive decline with normal aging as you get older. That's completely normal. However, over time, there's going to be this shift where the cognitive decline shifts to mild cognitive impairment, where there might be some symptoms that are present to the person and to the doctor, but it's not affecting everyday life. And then that will progress into dementia, both mild or mild and then to severe. There's that dotted line in the middle between preclinical and mild cognitive impairment. And so the preclinical stage actually defines the stage where some of these proteinopathies, such as beta amyloid and tau, are actually present in the brain. They can be measured with PET or potentially in plasma. But again, the person does not have any recognizable cognitive impairment to the person or the family. I, of course, could not go without mentioning Dr. Solomon Carter Fuller. He's the first African-American psychiatrist and he wrote the first comprehensive review of Alzheimer's disease. He actually worked in Dr. Alzheimer's lab, doing a lot of autopsy cases. And so a lot of the work that is actually, credited to Dr. Alzheimer's was also done by Dr. Fuller in this space. Because he was a person of color, wasn't given the same advantages, he was often given the work of doing autopsies. So in these autopsies, Dr. Fuller actually identified these senile plaques, and senile plaques is just another word for saying like these amyloid beta plaques in autopsy cases to identify. So we're going to also be looking at these trajectories that we talked about and how they function in Black individuals. So this here is looking at different biomarkers and their trajectory across the different clinical disease stages. So from normal, preclinical, MCI, and dementia, as we discussed. First, you can see that in the preclinical stage, I mentioned that there are some biomarker abnormalities. So for example, there's amyloid beta that is starting to become abnormal, and synaptic dysfunction, which can be measured via FDG PET, and then tau protein, lost my mouse, sorry, and tau protein here in green, which also starts to accumulate in the preclinical stage. The time scale isn't actually like mentioned on this slide, but it's important to note that a lot of these proteinopathies start to aggregate decades before any of the symptoms have begun to be apparent. Then in the MCI stage, you start to see brain structure abnormalities, for example, but in the hippocampus. And then in the MCI, you start to see cognitive changes, which can be measured via neuropsychological testing. And then you start to see clinical function in the later stages of MCI and dementia. Sorry, I'm looking this way because it's covered. So, so next, I wanted to ask, or the field has asked, is genetic risk for cognitive decline in AD the same for Black individuals? And in short, the answer is no. So oftentimes we identify genetic risk factors using GWAS or genome-wide association studies. So what you see here is significance on the y-axis, and you see different chromosomes on the x-axis. And this is called a Manhattan plot, because if you look at it, it's supposed to look like a Manhattan skyline. And the skyscrapers, or the really high peaks up here, are values that are deemed significant. So you see a well-known gene for Alzheimer's disease risk or dementia risk is ApoE, or apolipoprotein E, but you also see a variation of other genes that are actually not present in white individuals when looking at GWAS of Alzheimer's disease. The number of Black individuals for this GWAS is way, way less relative to the number of white individuals. So I think there's still work that needs to be done in the space in terms of recruitment and inclusive research. However, so far the evidence is leading toward the fact that genetic risk is different for Black individuals. I also want to follow up with some work by Dr. Elizabeth Rose Maeda. She's actually here at UCLA in Department of Epidemiology. So she did this great paper using data from Kaiser Permanente, people from Northern California, looking at dementia incidence rates by race and ethnicity. And as you can see, people who identify as African American or Black have a much higher incidence rate of Alzheimer's of dementia relative to other racial groups, especially relative to white, which goes to show again how important it is that we continue inclusive to better our practices of inclusive research in non-white individuals. So she also had created this really great health equity research goals. I'm a very big collaborator, and I really try to work really hard with people here who have similar missions. So you'll see with me, hopefully until I retire here, fingers crossed, hopefully really sharing a lot of the work that's being done here and building off of that as well. But a lot of the health equity research goals, we share a similar view. So we have this ultimate goal to identify drivers of racial, ethnic, and other social inequalities to inform policies and programs to promote health and equity. So that's the ultimate goal. But there, to get there requires a lot of individual research projects. So for example, describing health inequalities by social groups. Is this equality larger or smaller in different settings over time? Different settings being, for example, socioeconomic status, neighborhood disadvantage. And evaluate the extent to which particular factors contribute to health inequalities. So looking at these trajectories a little, looking at these biomarkers and markers a little bit closely, genetics is not the same, but another thing that's not the same is neuropsychological testing. Neuropsychological testing is created and validated in white participants and then given to people of all, given to everyone. And there are noticeable differences in performance based on race and ethnicity. So the first example I'm going to give, looking at this graph, on the y-axis, you have global cognitive score, higher is equal to a better performance. And then on the x-axis, you have age from 60 older, or that could be equivalent to time, for example. Highlighted in the green box is just one time point at baseline. And if you were to look at this person's, at this mean global cognitive score between white and Black people, it looks like Black individuals on average are doing worse relative to white individuals. But if you look at the slopes over time, you can see that they're not really different, suggesting that these neuropsychological tests are actually measuring what they're supposed to be measuring, cognitive performance. But if we look at them on one time point, because they're tested, these created and tested in white individuals, pretty much every other group is just not performing as well on this test. So that shows two things that are really important. First, the racial bias that exists in these exams, but also the need for longitudinal studies in Black cohorts in order to better assess cognition in these groups. So this leads to a study that I'm still working on this paper. And this is the Cogstate Computerized Cognitive Composite, or C3 battery. So there's a lot of, currently the neuropsychological tests are pen and paper tests. And there might be language proficiencies between ethnic and racial groups. There might be educational backgrounds that prevent people from performing as well as they should. So computer-based tests are a potential way of changing these things for the better. There was a recent paper by Gamato et al. That found older Black adults were more satisfied with computer-based tests compared to pen and pencil. And that's supposed to be pen and paper, but shown to these C3, these computerized tests were also shown to detect AD-related cognitive impairment, both cross-sectionally and longitudinally. And the C3 battery was associated with AD phenotypes like cerebrospinal fluid, hippocampal volume, and levels of amyloid, but only in white participants. In non-white participants, there's been no publications to my knowledge. So this led to the goal of the study of, does ethnic and racial bias exist for computerized neurocognitive battery? And this is important that I do this comparison because these computerized batteries are put out there as a suggestion that they are more culturally free tests. But I haven't seen a paper that directly compared them. So this C3 battery test actually uses playing cards, which is pretty cross-cultural to the paper's knowledge. So, and then here's the cognitive battery. So you have the C3 test, which includes both memory tasks, but also the CBB, or the cognitive brief battery, from visual learning, psychomotor, working memory, and attention. And the test measures vary based on what's shown here in the gray box. But for example, if you're doing the one back task, you want to mark yes if you've seen that card just one back. So if you flip a joker and then you flip a joker again, the participant should press yes, because that joker was just one back. So that's an example of how it works. It's just a yes and a no button for these cards. So for this study, I used data from the anti-amyloid and asymptomatic, or AD or A4 study that was launched in 2014. This is a secondary prevention trial in cognitively normal individuals that provides an opportunity to look at amyloid in a large data set of cognitively normal healthy controls. So again, we wanted to look at a C3 presence different across ethnic or racial groups. So this is looking at the estimate and the confidence intervals. You have different racial and ethnic group comparisons here: Black versus white, Hispanic versus white, and Asian American versus white. And I specified Asian American because this cohort actually has Japanese people, but I definitely believe that cultural influences have an effect on cognition. So I stuck with American group in particular. Anything that touches the dotted line is not significant. So anything that is to the right or left of this dotted line suggests a significant difference. And so just looking at this, you can see that there are differences in cognitive performance in what is supposed to be this less culturally biased test, still for Black individuals. This one was supposed to be over here, my bad, but these are still significantly different as well for all of these different tests. And I also want to just acknowledge Shinran Wang at UC San Diego, who helped me run a lot of these analyses. What is important to note, though, again, I did mention that education does have an impact on the test. And I do want to show that at least for most of these tests, that the education bias was reduced. I know I don't have it before, but trust me when I say education is usually a big predictor of cognitive performance. And so the fact that the estimate has moved so close to that non-significant line is important to note. So another thing about genetic risk factors, there is the ApoE4 gene, so apolipoprotein E4, that lies on chromosome 19. And there are 3 isoforms, E2, E3, and E4. And there is a dose-dependent effect such that 2 alleles of E4 has a higher risk than 1 allele of E4. And E2 is thought to be protective. However, because we do not have a lot of Black individuals that are present in our studies, E2 is also more rare. It is very hard to actually study or analyze the effect of E2 as a potential protective factor in Black individuals. So E4 is supposed to be this really large genetic risk factor for Alzheimer's disease or just overall cognitive performance. So what we have here is the percent of E4 on the y-axis, and then we have cognitively normal controls in the solid colors and people with dementia, Alzheimer's disease, in the pattern. So if you look at people who are cognitively normal, Black individuals actually have a much higher frequency of ApoE4 relative to other groups, including white individuals. So if E4 is supposed to be associated with this increased risk for dementia, then you would expect that more Black people who have this E4 are going to have dementia. But if you actually look at the E4 carriers with dementia in Black versus white, it's almost the same. This suggests that E4 is not carrying the same risk for dementia in Black individuals. Oops. Further suggesting that there are other factors, whether it be genetic or other things we're not focusing on, that are actually contributing to dementia risk in Black individuals. And there's a really great group out of University of Miami, Jeffrey Vance's group, that does really great translational work. I really hope to collaborate with him in the future. But essentially, they looked at ApoE4 expression in people with African local ancestry at ApoE4 and European local ancestry at ApoE4. So, and what you actually see is people with African local ancestry at the E4 allele actually have lower E4 expression relative to European people. And purple means more signal, more expression of E4, sorry. So you can see that there's evidence for actual low expression of this E4 in people of African descent. Another gene that I'm very interested in lies just upstream of ApoE, and that is TOM40. This encodes for a protein to allow a channel protein to allow proteins in and out of the mitochondria. Sorry. So there's a specific allele called the 523, or for short, that codes for intronic poly T repeats. So essentially, at this specific SNP or loci, you just have T, 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 T. And as you can see here from the counts, usually it's around 15 up to about 40 Ts at the specific loci. And this poly T repeat has been associated with increased cognitive impairment and Alzheimer's disease risk. However, it is in linkage disequilibrium with ApoE. Linkage disequilibrium is a non-random association of alleles. And so typically ApoE4 and TOM40 are inherited together. So previous research that I saw TOM40 in the signal just attributed it to ApoE. But guess where this research was done? In white individuals. I'm going to show you why that's important to note in just a second. So again, if we're looking at TOM40 in white individuals and Black individuals, the variation looks very similar. Just you see like a few people who have higher than 40 T repeats for people who identify as Black. But when we look at this in ApoE background, we can see that white E3 carriers had T alleles that were less than 19 or greater than 30. And on the ApoE4 background, they had T repeats that were between 20 and 30. So they call this short, long, and very long. So I don't have a clock, so if I get too long, just let me know, please. Sorry. Okay. Short, long, and very long. But if you look at Black people, the TOM40 variation across the E3 and E4, you see that Black E3 carriers have short and very long, but the Black E4 carriers have a much greater variation relative to white E4 carriers. And so again, when I talked on the previous slide about they attributed the TOM40 signal to ApoE4, it's because they were only looking at white individuals and there's no variation in white E4 carriers. Therefore, you really are capturing true linkage equilibrium between these two alleles. So I was really interested in what does this 523 or TOM40 variation in Black E4 carriers mean? And so for this study, I used data from Rush University in Chicago. And this has 3 different cohorts: religious order study, the Rush Memory and Aging Project, and the minority aging research study. And most of the data comes from the MARS study, which is Black individuals from the Chicago and surrounding suburbs. They have a really, really rich neuropsychological battery across different cognitive domains that I was able to look at. So again, we have these Black E4 carriers with variation. And so for statistical purposes, we had to group these. So we decided to group them by the presence of short. So if you have 2 short alleles, this is represented by red. If you have 1 short allele, this is represented by blue. I don't know, they're not the same color blue, sorry, but. And then if you have no short alleles, this is represented by gold. So what we see here is Black E4 carriers who do not carry the short allele had significantly faster rate of cognitive decline relative to Black E4 carriers who do carry the short allele. Now, again, when we bring in the white E4 carriers, remember there's no variation, so there's just 1 line, they're just Black individuals. First of all, we can see what I talked about in the beginning about the cultural bias that exists in neuropsychological testing. This is something that you see across every cohort with every neuropsychological test. And again, if we look at this baseline, you can see that 1 time point, it looks like white people are having a better performance in global memory relative to Black individuals. But if we look over time, and so this is a follow-up of an average about 8 years, we can see that white E4 carriers do not have a significantly rate of cognitive decline relative to Black E4 carriers who do not carry the short allele. However, Black E4 carriers who do have a short allele, this blue and red, do have a significantly slower rate of cognitive decline relative to the white E4 carriers. So this suggests that there's a potential a subset of Black E4 carriers that actually have a significant, like a potentially protective rate of cognitive decline relative to other Black E4 carriers and white E4 carriers. So yeah, again, here's another potential genetic risk factor that we're being missed if we're just attributing genetic risk markers that have been identified in only white people and trying to attribute it to Black people. Also important to note, while I looked at Tom 40, there are other non-APOE genetic risk factors that have been identified. Here is this other updated GWAS study that identified some novel common loci, novel rare loci, and then also identified other reported genetic risk factors in Black individuals. And importantly, when looking, this is that again from Jeffrey Vance's group in Miami. This is another version of kind of like a Manhattan plot, but they also found two SNPs that showed a significant impact for Alzheimer's disease risk. But they found that this one represented pink. They didn't actually list the SNP in the paper, they just had like a code. So this pink one had a lower AD risk for Black individuals, while the Black peak actually had higher AD risk for Black individuals. And this was based on ancestral background. So again, we're showing some potential new genetic markers when we focus solely on Black individuals. You've heard me talk a lot now about genetic ancestry, race, and I really wanted to take a moment to talk about how to appropriately talk about genetic ancestry in research. So race is a social factor with no biological relevance. I have started to try to say people racialize as Black. Have I said it in this talk? I don't think so. I'm working toward it. It's one of those things I'm trying to update my language based on, things we learn. And so I try to say people racialized as Asian, racialized as Black, racialized as, fill in the blank. And I think that's really important to note that really takes into context that it is a social fact. We are called Black based on other racial characteristics that other people have put on us. It's just a social construct. There is no biological relevance behind it. And I do think that race, and at the same time, race is important when we're doing studies to a point, because it does capture important epidemiological information. So for example, if we're looking at neighborhood disadvantage, lack of access to healthcare, again, this also goes to the social side of it, but advantages based on how people perceive you is a big part. So even though I say all these things against race, I do think it's important still sometimes in science, until we figure out a better way to do it, to still report race in this context for knowing how that might actually affect a person's health. Race does not equal genetic ancestry. Genetic ancestry is actually correlated with geographic location, which begs to another question that I'm not going to get into. Is it actually environmental? So genetic ancestry, if you're unfamiliar, they'll go to a specific region, let's just say, like Nigeria, and they'll ask a person, did both of your grandparents grow up here? And if you say yes, then you are Nigerian. They will take your blood, and then you're now in the catalog as Nigerian. We know, because of migration, this is not always the best way, right, to do this. Again, we're really focused on geographic location, not racial group. Also, lack of, that's not a mistake. I really mean a lack of, because they have not, there has not been enough efforts in African countries specifically to do this, to collect data for genetic ancestry catalogs. So these GWAS catalogs still are 80-something% European ancestry, and only 2% represent African background. And to calculate genetic ancestry, again, just to tie it all together, it is dependent on these reference panels that we create. So this is really important when we're considering things such as structural racism and discrimination, when we're talking about race, genetic ancestry. How can we merge all of these things together? Another thing that is not the same, you've actually heard me talk about so far, just amyloid and tau, but Postmortem neuropathological insults are not the same for Black individuals. So shown here in blue is the AD neuropathology, and so that is amyloid and tau. And you can see that for white participants, the AD on the right, the AD portion is a lot bigger relative to the AD portion for Black individuals. While other things such as Lewy bodies, infarcts, are more highly present in Black individuals. And this is true. Black individuals tend to have more comorbidities of disease relative to white individuals. So again, if we're only focused on two major proteins, amyloid and tau, knowing that this is not associated with the biggest risk, associated with disease in Black individuals, we're potentially missing a lot of other proteinopathies. And while the numbers look pretty low here, I just want to say this is just one of the first papers that came out. This has been replicated in some other larger studies, but again, not a lot of Black people in cohorts and not a lot of Black people that are going to donate their brain. So unfortunately, the numbers that we have compared to white individuals is not the same for this as well. And this is Lisa Barnes. She's my mentor and she's really great. So I wanted to highlight her work as well. So this is also shown looking at AD protein biomarkers in Black individuals. You could look at the years here, and you can see that only recently has there been a lot of work looking at protein biomarkers in Black individuals. And this is using fluid markers such as cerebrospinal fluid or CSF, or looking in vivo using amyloid or tau, PET or positron emission tomography. Again, it is typically not enough effort is put into recruitment for Black individuals for these efforts. But in general, you can see that typically tau individuals have lower tau, and that's pretty consistent between most of the studies. There's a few that have shown no difference, but of interest is amyloid, where there's quite a variation between study finding higher amyloid in Black individuals and or no difference in both CSF and PET. So there's these discordant amyloid findings. So again, I kind of mentioned that A4 study that had a lot of amyloid. So I actually have a data set that I'm able to investigate amyloid a little further. Unfortunately, to date, there is not a Black cohort that has tau PET. There are people that have just been working on this, but this is not available information to use. So in preclinical Alzheimer's disease, I mentioned before that amyloid already starts to increase, is already present in the brain and one of the ways that you can look at this is amyloid PET, and that is shown in that red arrow there. So in fact, people who are cognitively normal, about 30% of older adults, age 70 or older, actually have amyloid, are amyloid positive. So it's unknown, though, if these findings translate to non-white populations, that being that amyloid in cognitively normal individuals is associated with future memory decline and dementia. So first, we just wanted to look at the amyloid levels in Black and white individuals. So on the x-axis, you have continuous amyloid SUVR, which is just the amyloid measure. Higher amyloid is worse. So lower amyloid is better. And on the x-axis, you have the ApoE genotypes. And if you remember, it's like a docile effect. So E3 is what's present in the general population and is not associated with increased or decreased risk, while E4 is associated with increased risk for dementia. So, and then we have Black individuals in red and white and blue. So just by looking at this, you notice already that Black individuals have lower, AKA better amyloid relative to white individuals. But what's very interesting is that this was only significant, that Black have lower amyloid relative to white in the E4 carriers, which again, if E4 is supposed to be associated with increased risk and you're a Black E4 carrier, why only in this group do you have significantly lower amyloid? So here we, again, putting this all together, showing genetic and proteinopathies such as amyloid that are supposed to be this high risk, but we're showing this actually is equated with lower risk in Black individuals. So running out of time, just trying to think about how fast I should go. Okay. So let me just, I guess I'll try to cover this last part, but so is African ancestry associated with amyloid is another question I had. people who self-identify or have been racialized as Black in America have admixed genetic ancestry. This is people who have been here for generations. So this is due to the transatlantic slave trade, atrocities that were, done to our people in the past. So if you identify as Black in the United States, you typically have very admixed genetic ancestry between both African and European ancestry. And this ranges anywhere between 30 up to about 98%. In this study, the African ancestry is shown in red, and each of these stars is an individual person, but each person has 5 stars represented these 5 global population groups. So this person here who has about 28% African ancestry also has about 90% European ancestry. And so that's how you read that down. This has obviously been organized in increasing percentage of African ancestry, and you can see that the inverse of that is European ancestry, essentially. So African ancestry exists on a continuum in Black people from the United States who have been here for generations. So I wanted to look at this going along with the previous research, looking at potential ancestral background. And what I have here is amyloid again on the y-axis and African ancestry on the x-axis. And remember, lower amyloid is better. And as you can see, as you increase African ancestry, you actually have lower amyloid. And this effect that was actually independent of ApoE4, so independent. So here we have yet again another suggestion that these risk factors are different, may influence AD risk factors in Black individuals. You've heard me talk a lot about inclusion into trials, and so I'm going to go through this kind of quickly because I know I'm running out of time. The A4 study, I'm actually kept in all the people's data, even the people who did not get amyloid PET. If they did not get amyloid PET, that means that they failed screening and were excluded from the trial. So they could have been excluded for several reasons, whether it be medical, clinical dementia rating, mini mental state exam, or logical memory. And if you recall, what I've been repeating through this study is that neuropsychological testing is cultural bias. Even though there's some norming data that this group did, there is the potential that they have been kicking out people based on these cognitive test scores that should not be getting kicked out of excluded from the study. So indeed, for the A4 study, we saw that Black individuals were more likely to screen fail relative to white individuals. And why? Because Black individuals had too low of cognitive test scores. So again, if you remember that baseline, non-white individuals always perform lower relative to white individuals. Even if there's norming processes that are going on, we're still kicking out Black individuals because their cognitive test scores are too low, versus white individuals who were excluded because their test scores were too high. So I'm just going to run through this just to kind of sum up my work. I know I talked about other people's work as well, but just to sum up, ApoE4 is associated with cognitive decline, but in Black people, TOM40 seem to influence this effect. Black individuals have lower amyloid or better amyloid relative to white individuals. However, this is influenced by African ancestry, such that if you have higher African ancestry, you have lower amyloid. And Black individuals were more likely to screen fail out of the A4 study relative to white individuals. All of this actually leads me to believe, who are the Black people then that we're including in our study? If I've said now repeatedly that Black individuals, have this lower performance on cognitive tests and they're getting kicked out, that means that there are Black people that are performing at a higher than the average that are being included in these studies. So is it then that the Black individuals we actually have in our studies are this resilient Black group? Maybe there's other protective factors we're unaware of that are actually contributing to better performance. I do not know. And this is something that is of great importance to be looked at in the future. I'm going to just bypass this because I'm going through, I'm low on time. But the highlight of this is just Black individuals are more likely to show a lot of brain aging effects in middle age relative to late age relative to other ethnic and racial groups. One of the things that I have not touched on yet that I am now doing while here at UCLA is looking at non-biological factors. So environmental, sociocultural, and behavioral factors. And this is really, really important to look over the life course perspective, because it's not that all of a sudden at age 60, all of these things just happen to you and then you have cognitive decline. Things occur over the life course that impact cognition that are really important. We're going to skip that. And so I've been really focused on looking at structural racism and discrimination, neighborhood disadvantage. Experiencing racism throughout the life course causes stress. Stress can actually affect biological processes, which could then affect any aspect of health, honestly, right? Just in our case, I'm looking at dementia. And Dr. Paris Atkins Jackson, shown here, has been really great. She's also a new PI, a new faculty member at Columbia University. She's doing great work in leading people how to use structural racism and discrimination appropriately, ethically. And she's really been helping me out in this process as well. And an example of how we can attribute this racism stress biological processes, there is Dr. Laura Zahadny, and on the top you have hippocampal volume, lower is worse. And on the bottom you have white matter hyper intensity volume, and that is higher is worse. And so you can see that facing discrimination, increasing experiences of discrimination is actually associated with worse brain health for Black individuals. And this is data done only within Black individuals. And then I would like to kind of end on this health equity framework. I've again talked repeatedly about how we're not doing, and we being medical doctors, researchers, are not doing what we need to do in order to focus on the Black community. A lot of the research, it's geared toward white communities. There are cultural differences that just aren't being considered. And so, you lose a lot of participation that way. You're not able to communicate with them. So there are a lot of people who I've heard too many times that it's too hard when it comes to recruiting Black from the Black community, Hispanic, Latinx. And that's because, again, it makes me angry that when people say that, because you're not putting in the effort. You want to put in the effort to get a PhD, you want to put in the effort to learn imaging, to learn all these different techniques. But then all of a sudden, when it comes to actually, engaging with the community, that's too hard. I don't want to do that. I'm just going to stick with this. And then I hear these same people complain later, well, they're not coming in. They're not giving us, it just blows my mind. There are so many people out there, and actually not so many. There are very few people, but they are the ones who are putting in the work, that are publishing papers like this that are showing how to engage with these communities, and people are just ignoring it and still saying it's too hard. So I really hope that people that remain in this change that attitude a little bit and go read these papers and hire someone that can speak to the community directly, which I also plan to do here at UCLA. So yeah, I have now switched a lot of my research to focusing on within the Black community. Because again, if we continue to compare to the white individuals, you're always going to see a disparity, because every test, even the CSF and the PET, all of these levels of what we deem as high or low was validated in white people. So why research within the Black community is important. Black individuals are, disproportionately more likely to have cognitive impairment relative to white. Yet whiteness continues to be centralized in research. Again, here's an example of the GWAS catalog of genetics that I was talking about. 78% European and only 2% African, let alone all the other non-European groups. And this is really, really important because the United States is becoming more diverse. For the first time, we're actually seeing people who self-identify as just white on the decline, while every other group is on the incline. So personalized medicine, looking at genetic background, is going to be really, really important in the future. So this brings me to my lab. I now have a lab logo. So my lab really takes into account not only biological factors, but now cultural influences. How do social factors, how do biology, how do they interact or not interact to influence brain aging and ultimately dementia and Alzheimer's disease? And so with that, I've been here for like 7 months. So a lot of this is just acknowledgements for other places I've been and past funding. So hopefully this UCLA group will grow over time. And yeah, with that, thank you for listening, and I'll take any questions.
There is time for questions, both online and in person. So if you are online, just type your questions into the chat. It should be enabled. And if you're in the room, we have 2 microphones. We can run around and grab you. So.
Hi, Kacie. Great talk. I have a question about the relationship between APOE and cognitive decline, particularly, between racial groups. I was curious if you have examined or found any kind of interactions that jump out at you. Maybe gender is one I'm really interested in, or any specific lifestyle factors which may interact with those differences that you showed.
Mm-hmm. Yeah, so great question. So another downside to the fact that there are less Black individuals in these cohort studies is there's also a major gender imbalance. There are significantly more Black women in studies than there are Black men. It has actually made this extremely hard to look at gender differences in the Black community. To my knowledge, there is one group that is looking at this by aggregating across different cohorts. But as we know, a Black group from LA is not going to be the same as a Black group from Chicago, so it's really actually hard to aggregate across cohorts. So it's a good question, but unfortunately, to my knowledge, we don't know the answer to that yet.
Thank you. Great.
Yeah.
Thank you for that wonderful talk. This is super loud. I was wondering about the baseline differences that you sort of mentioned again and again. How much of that is, as you just sort of alluded to, sort of selection related of the people who actually are included, and how much of it, so you said, oh, the computerized design is a little bit, shows less differences there. So how much is still related to design issues of the questions of those?
Yeah, so I am not a neuropsychologist, although I pretend to be. I just went to a conference and they said I could be an honorary neuropsychologist. So I don't know all of the details about like that type of facet. One thing I do know about the test is that a lot of the things in the test were, I like to use the Boston naming test as an example, where pictures are shown and then the person is supposed to identify the picture. And so a lot of these things were really from like the East Coast, for example, or they're so old it's from stuff that just no longer is around. For example, there was like a picture of a hoe, like for farming. Like people don't really use that anymore, and so people don't know how to use it or won't know how to say it. Additionally, up until 2 years ago, there was a noose on the BNT. Oh. You can imagine the stress for not only Black individuals, and literally up until 2 years ago. And I think some people still might decide to use it, but not only is stress for the Black individual, but even white individuals were like, people would be like, I refuse to say that, as they should, just like people. So I think that there are just so many different factors that actually go into these neuropsychological tests that I don't fully know how to answer that question, but that's what I got.
It just seems really important because if you're comparing fundamentally different segments of the population that were selected from a larger group to different extents, of course you're going to get different baseline values, right? Yeah,
and so these cohorts are also very different too. Like the A4 cohort I talked about is actually a clinical study, and so these people were recruited from typically hospitals or other care centers, memory centers, and so these are people that clearly already have access to healthcare, but also will go in and are willing to participate. So there's that willingness that's already there. Versus the Rush study that I talked about, those are mainly community dwelling older adults. There is still a bias about where you go, of course, for these things, but these were people from, they went to churches, community centers, senior homes, things like that in order to recruit. So they were not recruited from the younger community. So I agree.
Hi, you have a great question online. Sarah Dunlap asked, she said, excellent talk. I was wondering if you or any other group have looked into postmortem tissue to explore regional anatomical differences in TOM40 across ethnic racial groups and how that might relate to cognitive resilience or decline.
Thanks for the question, Sarah. No, to my knowledge, there are like two groups, me and the guy from Miami, that are looking at Tom40. I think there's one more, but I'm not sure that they're actively studying Tom40. It is, it just really hasn't, it's been ignored. It hasn't been funded. Because again, that association I talked about with of linkage to equilibrium with Tom40 and ApoE, that was only present in white individuals. Yeah, it's just it's been largely ignored. So to my knowledge, we're the only two groups, and we don't have postmortem data on this right now. It is a great idea. Also, it's been brought up, multiple times. That, again, I don't have the data to do this, but hopefully we can collect something in the future. But obviously, it's a mitochondrial protein, function protein. So look at mitochondrial dysfunction in these individuals as well. Could that be a potential mediating factor? So.
I also have a question. And Carrie Bearden says, thank you, Dr. Deters, for a great and inspiring talk.
You're welcome. Thank you.
So my question is, have you thought about looking into epigenetic factors?
I have actually. That was a whole passion of mine, my PhD. And then I had to say, stop, Kacie, otherwise you'll never graduate. And so I'm actually very interested in epigenetic changes, especially when it does come to environmental factors of people, typically minoritized or racialized individuals, have been redlined, and therefore there's other factors, environmental factors. I think that that's very interesting. I think that that could be a project if someone wanted to join my lab in the future. But yeah, I think people are starting to look at that, and I would love to, but I don't have the bandwidth right now. It's always in my mind, like always. I really hope that I can incorporate it, but also there's just also not a lot of data on that either right now for Black individuals. So yeah, very data restricted.
Oh, we have another question, Roy.
Great talk. Great talk. Great lab logo. I love it.
Thank you.
I had a question about how do you turn some of these genetic risk factors that have been identified and convert that into, I guess, like basic science, and how do we start approaching mechanisms of potential risk factors?
Yeah, I got where you're going. Yeah, it's a great question. I really hope for my work to be translational one day. I think that's one of the reasons actually why I know that's actually one of the reasons why I joined UCLA in this department as well. Identifying genetic risk variants and being able to express these things in like a mouse or a fly and look directly at the implication of what does this genetic mutation do. However, this is not like my background field, so I may be saying this wrong, but also if it's possible to, that is not just one genetic factor. First of all, it's contributing to dementia. There's got a whole host. So if it's possible to start looking at the interaction or doing multiple genetic mutations in a model and looking at the ensuing phenotype, I think it's going to be really important, and somewhere I definitely hope I go with my research, because human research is great, but it is association and translation. So being able to determine what is the actual impact of this is really important. And again, that's also, I talk so highly of Jeffrey Vance's lab, but I really love what they do, because they find something and they try to look at what does this mean translationally using like iPSCs, for example. So yeah, great question.
Oh,
I'm kind of scared of this microphone. So I've been thinking about how to like formulate this question in an intelligible way, and I guess it's mainly a question about how you view like a distant future. If we're optimistic and we think that down the road maybe we're going to have improvements in diversifying human research.
Mm-hmm.
I'm curious about whether you think that, that work is obviously important to make sure that we're coming up with healthcare that's equitable across all human patients. But do you also feel like there's this opportunity embedded in there to maybe like find factors that contribute to resilience in a way? Like we're like as a research community, we're missing opportunities to find some factors for like resilience and protection. The thing that you spoke about that really was planting this seed in my mind was just the observation about the lower amyloid levels, but like only when you also are an E4 carrier.
Mm-hmm.
Like that's really intriguing, and is a finding that is not sort of complicated by the neuropsychological tests and their challenges. So I don't know, I'm just curious about whether you also are maybe motivated by there being this potential opportunity for us to find new factors of resilience by having more diverse populations in the human research space.
And Mike, no, absolutely. And there's, a whole field of people. Actually, I unfortunately bypassed your slide. This may have actually answered part of it, but like Dr. Crystal Glover is an example from Rush University, who looks at, resilience factors. And Laura Zahodny actually showed some of her work, but she's somebody who also looks at resilience factors. So there are groups of people that are looking at this as well, in the opposite way. I do think it's we're missing a lot, first of all, by not diversifying our sample. We're missing a lot. I hope one day that we don't have to, segregate our studies based on racialized groups. But the way America is structured, we know how much life course social factors, the policies that are in place have an effect on healthcare. So until policies change and that it changes our overall, healthcare, and overall health, I don't know that, that's going to be doable. But yeah, resilience factors, I think is really important to look at, because we look at everything as a risk factor rather than resilience. I feel like I'm kind of stumbling around the question because I'm like also trying to think a little bit about about that. But yeah, yeah, I'm not sure I would have anything else. Yeah. Yeah, I think, identifying and focusing on those is going to be important in the future, but I'm not sure how to focus on that in a population that already has so much disadvantage, if that makes sense. Yeah.
I think research like this could actually cause policy to change and make the difference, the long-term difference. So keep doing what you're doing. And I just want to thank everybody for coming, and thank you online. And thank you, Dr. Deters, for an amazing Martin Luther King and Black History Month talk.